All right, well, hello everyone. So we are starting Unit 2 today, taking a look at DNA. So that's what will be our first set of notes here for Unit 2 or DNA. So I like to, start a whole new chapter or whole new unit here. So I'm gonna go all the way up to the top, fresh page, Unit 2, and this unit is called nucleic acids. In our first section, 2.1. is the structure of DNA. Now, perhaps yesterday when we did the Hershey and Chase experiment booklets, perhaps before that, you guys were unaware that the NA in DNA stands for nucleic acids. There's our N, there's our A. Nucleic acids. So again, we did the activity yesterday. I think you guys all knew that DNA is the genetic material of life, but the booklet always talked about nucleic acids and not directly DNA. So that's what the NA and nucleic acids stands for. So if we're starting about the structure of DNA, we have to start at its what we call monomer unit. And that's the nucleotide. So monomers, we're going to see a lot of these over the next couple of chapters. Monomers are kind of like train cars. So you guys can all picture a train that's made up of a whole bunch of individual cars. And if you have just one of those train cars, it's not a train. You have to link a bunch of them together to make the train. Well, the same is true of our macromolecules. So nucleic acids are the macromolecule, and you link a whole bunch of the nucleotides together to make a nucleic acid. So these are the train cars. This is the train. So starting off with the nucleotide, and all nucleotides are made up of three parts. Phosphate group, a 5-carbon sugar. We'll talk more about that in just a second. In the case of DNA specifically, that 5-carbon sugar is called deoxyribose. And there's the D for DNA. So deoxyribonucleic acid. That's our DNA. There's where the word comes from. So here's our 5-carbon sugar. Notice it's drawn kind of in a pentagon. That's true. In chemistry, we oftentimes use shapes like this, and every corner represents a carbon. So here's the first carbon. Here's the second carbon. Here's the third carbon. Here's the fourth carbon. And here, kind of up here like a flag, kind of sticking off on the end, is the 5th carbon. You notice I put an O at the top of the pentagon, and that's because it's an oxygen at this corner, not a carbon. It's an oxygen that is holding that ring structure together. The 5th carbon kind of sticks off here like a flag, and that's what connects to the phosphate. And finally then, coming off of the 5-carbon sugar is what we call a nitrogenous base. Now, nitrogenous is a funny sounding word. You don't hear it very often. What it really means though is just full of nitrogen. So OUS means full of. So this is full of nitrogen. We'll take a closer look at that in a minute. Not that it's incredibly important, but for right now, there are 4 bases to make sure you know. First one is adenine. Second one is guanine. It's a U there after the G. Sorry for that. Thymine and cytosine. Those are the 4 bases. And most of the time, you're perfectly welcome to just use their 4 one-letter abbreviations. So we talk about the A's, the T's, the C's, and the G's. of DNA. And that's what we're talking about. So I want you to make sure you write them down now so you can have them, so you can see them, because sometimes you will see the full word written out. But more often than not, we're just going to be looking at that single letter abbreviation. Back to our nucleotides here then. So here's the 3 parts of every nucleotide: a phosphate group, 5-carbon sugar, and a nitrogenous base. The phosphate group and the sugar are always the same. They never change. That part of the molecule is always the same. What can change is the base. You can have an A, you could have a G, you could have a T, or you could have a C. You could have any one of those 4 letters representing your base. So that's what changes as you move through the different types of nucleotides. I'm going to kind of blow this up because we're going to talk just a little bit more about our nitrogenous bases. There are 2 types. The first type is called a purine. They have 2 ring structures. So this is what they look like. Don't memorize this. It's not something I would ask. What's important is that purines have 2 ring structures. That part you need to know. Ultimately, here's what they look like. Again, the corners always represent carbons. There's a lot of carbon here, but the corners that have been replaced with an N are nitrogen. Not important. You don't need to know about that. I just want you to see it. So, hey, you can see it's called a nitrogenous base. It's a base that's full of nitrogen. So the 2 purines are A, adenine, and G, guanine. Those are the 2 purines. We also have pyrimidines. And the difference is that these only have a 1 ring structure. So they look a little bit more like this. Just 1 ring. So we've got the double ring, and this one only has a single ring. Again, I've got the nitrogens in there just so you can see that it is a nitrogenous base. It's a base full of nitrogen. And these are, well, the other 2 we have left, C and T, cytosine, thymine. So you notice how the purines are bigger than the pyrimidines, a little bit longer. That's going to play a really important role in a little bit here. Again, don't memorize the overall structure. You don't have to draw that, but you do have to know that purines are 2 rings, and so they're longer than pyrimidines, which only have the single ring. A and G are purines, C and T are the pyrimidines. All right, so let's start linking some nucleotides together. So here's my first nucleotide. Now we always start with that 1' carbon. That first carbon always connects to the base. So here's our base, there's our first carbon. It's connected to the base. And yes, it is going to be important to know the numbering system. So again, the first carbon always connects to the base, and then you just move clockwise and count the corners. Don't forget 5 sticks up like a little flag outside of here. So there's our first 1' carbon, 2' carbon, 3' carbon. Those are going to be super important. 4' and 5'. So let's start linking these nucleotides together. I'm just going to draw a dotted line between this one. This is the link we're going to be looking at. That's what some people have asked. Hey, did you forget to add the second box on this one? Nope, not necessarily. Now, you could have added one. That's absolutely fine. But I'm showing you the difference between a purine and a pyrimidine. All right, so here we have 3 nucleotides, and they're all linked together. And here's why knowing that numbering system is so very, very important. We can only link them in this direction. 5' to 3'. It's the only way it works. You can't back this one in 3' to 5'. It just doesn't work that way. It always has to be 5' to 3'. Important today, and that's why you need to know the numbers, but that's going to be really, really important when we get to DNA replication in a couple days here. So make sure always linking 5' to 3'. All right, we put a couple of stars here next to bonds. So the bond between this 5' and the phosphate. This one eventually will be. This one here is. This one here is. And this one here is. All these stars, these represent phosphodiester bonds. This is a special type of covalent bond between the phosphate and the sugar. So between the phosphate and the sugar, we're forming a phosphodiester bond here. Here's a phosphodiester bond. There's again, that's the 5' carbon, the flag of the sugar is linked to the phosphate with a phosphodiester bond. Phosphodiester bond, phosphodiester bond. So an important word, I'm guessing you probably see it like once more on the test. So if we're talking about 1 point here. So if you want to completely forget about it, it's too big of a word for you, that's fine. You'll miss 1 point, but at the end of the day, you can still probably get an A on the test without ever seeing that word again. On the other hand, if you're going for those really high scores, you want to make sure you see that you know this word because you will see it again. One more thing to add to this though, as we link 5' to 3', we're losing a water molecule. I actually thought about having you guys draw the C's and the O's, the P's. If you guys remember, we actually drew a phosphate group when we did our, when we linked our fatty acids to our glycerols last chapter. So it's the same thing, same phosphate group. If you remember, it's got these O's coming off of it. So it's really got like 4 O's, one here and one there. Thought about having you guys draw that. I'm like, not the biggest thing in the world. Would have been easier to see though, that the OH from the sugar and then H from the phosphate are linking together and coming off and taking their spot. So losing water, taking their spot would be this bond forms. So it's just like linking those glycerols to those fatty acids, a dehydration synthesis. Water is lost during the process. It's a dehydration synthesis. All right, you might have more room than I do left on your page. I still advise flipping over. We actually have a pretty big diagram to get going with here. We're going to now draw the double-stranded DNA. So I've got 2 purines, 1 pyrimidine. Not that that's terribly important yet, but here in a few minutes it will be. I'm going to label my 5' and 3'. Again, those are the big ones. The 1', 2', 4', not terribly important for this class, but they do help us think about where the 3 and the 5 should be. So if you're adding them to every structure you have, you're probably going to get comfortable with that really quick. So as we mentioned earlier, this part here, always the same. It always goes phosphate, sugar, phosphate, sugar, phosphate, very repetitive, nothing changes. Phosphate, sugar, phosphate, sugar, phosphate, sugar. This is actually what we call the phosphate sugar backbone. Think of a zipper, and this is the part of the zipper that connects to your jacket. It's kind of the straight solid line, and then you've got these teeth that come off of it. This is one strand of DNA. So you're looking at one strand. It's not on the camera there. So here's one strand of DNA. Here's what I'm going to advise you to do, though. We're going to draw the second strand. I advise you to flip your paper upside down. Forward, I had to do this upside down, and it is not fun. Not at all fun. There is a very good reason for flipping it upside down. We'll talk about that in a minute. Boy, it'd be helpful if I could actually get these lined up, huh? So kind of have to pretend that that's in a better line. All right, there we go. And again, for good practice, probably want to add our 5 primes and 3 primes. Those are the important ones. There we go. And we notice every time I had a pyrimidine on the opposite side, I have a purine. Purine, pyrimidine, purine, pyrimidine, pyrimidine, purine. That's going to help us keep everything level. We'll kind of talk about that in just a second here. So here is a second strand of DNA. And every time you have a first strand, you're going to have a second strand. So like your zipper, you've got your 2 halves. You've got the side on this side of the jacket and the side on this side of the jacket. Unlike your zipper, DNA doesn't very often come unzipped. You know, you can wear your jacket around with just one zipper up. We will unzip DNA, but it goes back together pretty quick when you do unzip it. So here's our second strand, and it's pretty much always going to be found with that first strand. This is what's called double-stranded. You hear that a lot. DNA is double-stranded, and here's why. It's like a zipper that you always have zipped up. It's got 2 halves that link together in the middle there. Why do we turn that paper upside down? Well, those 2 strands. Wrong page here. Those 2 strands are what we call anti-parallel. You guys all know the word parallel. 2 things are the same. 2 things that are running in the same direction, 2 parallel lines like railroad tracks. Well, anti-parallel means they are parallel, but they're moving in opposite directions. Good way to show that, up arrow, down arrow. They're parallel, but they're moving in opposite directions. And that's why we drew one upside down and one right side up, because they're moving in opposite directions. They're anti-parallel. And that's the only way that these pieces will fit together. So the bases, remember, these are the bases. And the bases pair up in the middle with hydrogen bonds. So remember those from last chapter, H bonds or hydrogen bonds? That's where they pair up. And there's some rules to that. They're called Chargaff's rules. A always pairs with T. So A always goes with T. And between them are 2 hydrogen bonds. Let's go up here. Let's say we called this an A. If this were an A, this one here would be a T. I've got it upside down because remember, that strand's moving upside down. And between them, we have 2 hydrogen bonds, linking them together. The other rule is that C and G pair up. And between them are 3 hydrogen bonds. Again, there's a bit of memorization here. I think this one's pretty easy. A and T, the capital letters, are always made with straight lines. So the straight lines go together, C and G are made with curved lines. The curved lines always go together. So back up to here, this one here, we can name it either T or C, not A or G. This one is a shorter pyrimidine, so it has to be C or T. If I call this C, then okay, that works. That means over here on the other side, it's a G. Again, I got that upside down because it's running in the opposite direction. And between them will be 3 hydrogen bonds. Finally, back to this one. Again, this is a pyrimidine. It's longer. So it has to be either A or G. I'm going to call this one G. And that means over here must be C. Again, they're going to have 3 hydrogen bonds between them. Now, the number of hydrogen bonds between them isn't really that big of a deal. I just think it's kind of cool. For instance, we talked about those thermidacidophiles from last chapter with bacteria that live in really, really hot springs. As it turns out, their DNA is almost entirely made of C's and G's. That way it can hold together better in those extreme temperatures. It's a little stronger bond between the C's and the G's than the A's and the T's. Just one more hydrogen bond making just the slightest of differences. So notice how we always have a purine pairing with a pyrimidine. And I mentioned that already. And the reason we have that, and the reason why it's so important, is if you had, if you had 2 long ones go together, 2 purines go together, it'd be too wide. And vice versa, if you had 2 pyrimidines go together, it'd be too short. Versus 1 long and 1 short, always a consistent width. So see, this is too long, too wide, too narrow. But this is always just right. As long as you always have a 2 and a 1, you're always going to have 3 bases across. You're always going to have that consistent width on your DNA. And that's going to be really important as we continue to build these long strands of DNA. All right, finishing up. The final thing that happens here is that our 2 strands were double-stranded, and they twist into a helix. Hold this up, here's my DNA, and it's twisted into a helix. If you straighten it out, mine's a little stiff, it's been like this for a couple of years, but it's twisted. There we go, it's not too bad. It's twisted around into a helix. And that's where we get the name, the double helix. 2 strands makes it double, helix because it twists. There's our double helix. That's that image you're probably very familiar with, you've probably seen it before. Kind of, we'll recognize that image of DNA. All right, well, there's our first bit of notes here in Unit 2, DNA, so take a look at those. We did do an activity. We actually built these models of DNA in class. There's really no way for me to have you guys do it at home. So if you're at home and you'd like to do it, like to participate, like to get this lab done, when you come back, talk to me about coming in during pathways, and hopefully we can work out a time for you guys to come in and build that activity. But otherwise, I think it'd probably make you kind of excused from that. But until then, until next time, see ya.